Hello viewers, welcome back to our channel, and we hope you have gone through our previous episode where we discussed about the neuronal structure, and by now we know that a neuron can connect with another effector cell, which can be another neuron, a muscle cell or a gland. This connection is brought about by some specialized junctions called the synapse. In this episode, we will try to understand these specialized communications called synapse. A synaptic connection is typically found between the terminal part of a neuron, which is usually its axon terminal, and the initial part of another neuron, which is usually a dendrite. These connections happens across a gap which lies in between the axon terminal and the dendrite, known as the synaptic cleft. Depending upon the nature of the materials which is being exchanged between these two neurons in a synapse, the synapse may be classified into two types: electrical or chemical. In an electrical synapse, there is exchange of ions which happens by gap junctions. These gap junctions contains channels that cross the plasma membranes of both the cells. The pore of these gap junction channels are wide enough to allow ions and even medium-sized signaling molecules to flow from one cell to the other. This is essentially a connection in between the cytoplasm of the two cells. Therefore, when the membrane potential of one cell changes, the ions move from one cell to the next, carrying positive charge with them, thus depolarizing the postsynaptic cell. In this way, in an electrical synapse, the conduction becomes faster, but the electrical response in the effector cell is smaller in amplitude.
Now, a chemical synapse, on the other hand, uses messenger molecules or ligands housed in some vesicular structures called the synaptic vesicles to be released into the synaptic cleft. Most synapses in mammals are of chemical type with a few exceptions, like retina, cerebral cortex, and brainstem, where electrical synapses are found. In this episode, we will be discussing about the chemical synapse. The messenger molecules in a chemical synapse are of two types, namely neurotransmitters and neuromodulators. These molecules are released from the axon terminal to depolarize or hyperpolarize the membrane of the effector cell. Within our CNS, there may be several types of possible synaptic contexts depending upon the regions of the neurons involved. They can be exoaxonic, exosomatic, exodendritic, or dendrodendritic. The most common of them all is the exodendritic type, where the axon of one neuron synapses with the dendrite of another neuron. In all chemical synapses, the message travels across the synaptic cleft to the effector cell, giving rise to some terminologies like the presynaptic cell, which is the neuron releasing the messenger molecule, and the postsynaptic cell, which is the neuron or the cell which is receiving the messenger molecules. This cell is also called the effector cell. Thus, if we again look at the major functional components of a synapse, there are three, namely the presynaptic membrane, the synaptic cleft, and the postsynaptic membrane. Now, these membranes are specialized so that the messenger molecules contained within the synaptic vesicles are released at the proper active sites on the presynaptic membrane, and so they bind to receptor molecules located at specific sites of the postsynaptic membrane. When seen in electron micrographs, the cytoplasmic aspects of either one of these membranes frequently display a filamentous electron density known as the presynaptic density and the postsynaptic density. Depending on the arrangement of these submembrane densities, the synapses are said to be either type 1 or type 2. The type 1 synapses are also known as asymmetric synapses, since their postsynaptic density is thicker than their presynaptic density. The type 2 synapses are also referred to as symmetric synapses since the two densities are of equal thickness. In general, the type 1 synapses are excitatory so that the postsynaptic membrane of the effector cell becomes depolarized, initiating an excitatory postsynaptic potential or EPSP. Conversely, the type 2 synapses are usually inhibitory, initiating an inhibitory postsynaptic potential or IPSP so that the membrane of the effector cell becomes hyperpolarized, causing it to become less responsive to attempts at depolarization.
As mentioned earlier, the presynaptic membrane, the synaptic cleft and the postsynaptic membrane are the major functional component of a synapse. So let us have a look at these functional elements one by one. The presynaptic membrane is the area of the plasma lemma of the axon terminal that contacts the synaptic cleft. When we look at the cytoplasm of an axon terminal, it is rich in cytoskeletal elements such as microtubules, neurofilaments, spectrin, and microfilaments. Additionally, it also houses cisternae of smooth endoplasmic reticulum, coated vesicles, occasional endosomes, a number of mitochondria, and numerous synaptic vesicles. Synaptic vesicles are membrane-bound vesicles, the membranes of which contains numerous unique proteins that assist in binding the vesicles to specific site within the axon terminal. The synaptic vesicles may contain chemical messengers which can be a ligand or a neurotransmitter molecule. They may contain ATP molecules or some proteoglycans. All of these are destined to be released into the synaptic cleft. Another important component in the presynaptic membrane are some oblong structures made up of protein complexes known as docking complexes. These complexes form a grid-like network called the presynaptic grid and contributes to the submembrane density on the cytoplasmic aspect of the presynaptic membrane. The interstices of this presynaptic grid are just large enough to accept the synaptic vesicles but prevent these vesicles from contacting the presynaptic membrane. When an action potential arrives at the axon terminal, some synaptic vesicles are bound or docked to the docking complex. Thus, they are ready to fuse with the presynaptic membrane, open into the synaptic cleft, and release their messenger molecules. A mechanism very similar to loading and unloading of goods after a ship is docked in a docking area of a port. However, all the synaptic vesicles are not always docked but are mostly kept in reserve away from the active sites. Let us elaborate the process further. Arrival of an action potential at the presynaptic membrane opens voltage-gated calcium channels located at the active site, possibly as a part of docking complex, which permits the influx of calcium ions into the axon terminal. The presence of calcium ions, in turn, permits the fusion of the membrane of the docked synaptic vesicles with the presynaptic membrane and the subsequent release of its messenger molecules into the synaptic cleft. Now, precise control of calcium availability is necessary to ensure that only the required amount of messenger molecules are released as the consequence of a single action potential. After an action potential is over, further release of neurotransmitter is prevented by reducing the calcium level in the axon terminal. This is done by three mechanisms. First, the cytosolic calcium ions are actively exchanged for sodium. Secondly, it is done by sequestering the calcium ions in the smooth endoplasmic reticulum. Thirdly, by binding the calcium ions to some cytoplasmic proteins. Another important mechanism exists to check the continued fusion and incorporation of the docked synaptic vesicles into the presynaptic membrane, because if this is allowed to happen, it would enlarge the axon terminal beyond the normal limit. Therefore, this excess membrane of the synaptic vesicles are recaptured by the formation of some clathrine-coated pits. The pits form clathrine-coated vesicles which travel as endocytic vesicles, possibly along microtubule marked pathways, and the captured membrane are thus returned to the cytosol. These vesicles lose their clathrine coat before fusing with the elements of smooth endoplasmic reticulum to be recycled in the formation of new synaptic vesicles. All this above was an explanation of how an excitatory postsynaptic potential is initiated in a synapse, and this mostly happens in type 1 synapse. And since the flow of calcium ions into the axon terminal is very important for the release of the messenger molecules into the synaptic cleft, a method of diminishing its entry into the end foot has evolved in mammals, a process known as presynaptic inhibition. This process occurs in areas where an axon forms a synapse with the axon terminal of another neuron. There are 3 neurons involved at this location. The axon terminal of the first neuron releases a chemical messenger destined for chloride ion channels of the axon terminals of the second neuron. The binding of the ligands opens the chloride ion channels, causing an influx of chloride ion into the axon terminal of the second neuron, thus hyperpolarizing its presynaptic membrane and making it much more difficult to open its voltage-gated calcium ion channels. A very similar kind of mechanism of opening chloride ion channels happens in a synaptic inhibition, while an inhibitory postsynaptic potential is initiated in type 2 synapse.
We hope by now the anatomy and the functioning of an axon terminus and the presynaptic membrane is understood. So let us move a little more and discuss the synaptic cleft and the postsynaptic membrane. By now we know that the space located between the presynaptic membrane and the postsynaptic membrane is known as the synaptic cleft. This extracellular space is about 20 to 30 nanometer wide. This space contains diverse families of macromolecules, some of which appear to be filamentous and span the entire width of the synaptic cleft. Chemical messenger molecules in the form of neurotransmitters are released into the synaptic cleft in predetermined quantities. As many as 20,000 molecules of these neurotransmitter substances is called a quanta. These molecules cross the synaptic cleft and bind to specific receptor molecules on the postsynaptic membrane. Each synaptic vesicle contains 1 quantum of chemical messenger molecules, which is the minimum amount necessary to precipitate a single postsynaptic event, either a depolarization or a hyperpolarization. The postsynaptic membrane is a region of the effector cell plasma lemma that is modified to receive and react to the neurotransmitters released into the synaptic cleft. Binding of these neurotransmitter substances results in the opening of ion channels with subsequent depolarization or hyperpolarization of the effector cell plasma lemma. The postsynaptic membrane houses 2 major types of receptors, ionotropic and metabotropic receptors. The ionotropic receptors are ligand-gated ion channels, and the chemical messengers that bind to them are known as neurotransmitters. The binding of a neurotransmitter alters the conformation of the protein constituting the ion channel, opening it and permitting the flow of ions through it. Thus, a neurotransmitter is a chemical messenger that has a direct effect on the ion channel, and the effect is rapid. Metabotropic receptors bind chemical messengers known as neuromodulators or neurohormones. These molecules also interact with a receptor molecule, causing a conformational change in its protein structure. However, the receptor molecule is not an ion channel. Instead, this receptor molecule interacts with other protein complexes such as G proteins. The G proteins in turn interact with one of the cell's secondary messenger systems, thus modulating the metabolic activities of the effector cell. In this fashion, the required response occurs in an indirect function of the coupling of the ligand to the receptor, and the effect is slow and widespread. Thus, the neurotransmitters have a direct influence, whereas the neuromodulators or neurohormones have an indirect influence in affecting the desired result. It may be noted that many chemical messengers that are neurotransmitters also act as neuromodulators. Therefore, acetylcholine, dopamine, histamine, norepinephrine, and serotonin can be both neurotransmitters as well as neuromodulators. The difference lies in the receptor molecules on which they act. If they act on an ionic channel, they are neurotransmitters, and if they act on the metabotropic receptors, which in turn influences a secondary messenger, then they can be considered as neuromodulators. So folks, that's all we have for you today about synapses. Stay tuned to Anatomy Weekly. Keep watching, keep learning. We'll see you next time. Thank you.